Now, just to recap as far as histology, this began on page D1. And we learned that the cells, these early embryonic stem cells, initially have the potential to grow into any tissue or any organ of the body, but they undergo differentiation. The word differentiation sounds like the word different. They become specialized. And they eventually, first they specialize into ectodermal cells and mesodermal cells and endodermal cells, and they progressively specialize even more. And eventually they become liver cells and fat cells and nerve cells and so on. At the bottom of D1, we learned that there are 4 categories of tissues in the human body: epithelial, connective, muscle, and nerve. And all the organs of our body are made up of 2 or more tissues. That's the definition of an organ. You'd say, like, should we know what the definition of an organ is? Absolutely. You should know what an organ is. So on page D2, we started talking about epithelial tissues. And epithelial tissues are characterized by being made up of cells that are packed closely together. Curiously, there are no blood vessels within the epithelial tissue. And because of that, we explained that epithelial tissues, therefore, are always next to a connective tissue, which is vascularized. You'd say, wait, when did you tell us that? It's not written here. We showed you at the bottom of the page. We said this is an epithelial tissue, and it's resting right on top of a connective tissue, which is vascularized. Obviously, we'll learn more about connective tissues yet today. So we further categorize epithelial tissues based upon whether there's one cell layer thick or multiple cell layers thick. If it's one cell layer thick, it's called a simple epithelium. If it's multiple, it's called a stratified epithelium. Secondly, we categorize them, classify them based on the shape of the cells. Are they round and flat or squamous shape? Are they cuboidal shaped, or are they tall columnar shaped cells? We also learn on page D3 that some epithelial tissues, especially the columnar shaped cells, may be modified. They may have cilia. They may have microvilli. Which one move, cilia or microvilli? Cilia. Microvilli don't move. Then what's the purpose of microvilli? Increase surface area for absorption. So you'd say, did you write that? It's written right here. Yep. And then we mentioned mucus-secreting goblet cells. This is just a vacuole that's releasing mucus. And we have a lot of mucus-secreting cells all over our body. So we started to enumerate some examples of epithelial tissue. These are covered in your textbook. They probably mentioned 30 different types of epithelial tissue. In your lab manual, they probably mentioned 20. I think I gave you about 10. So the first was simple squamous epithelium. It basically looks like this. It can be folded or rolled into a tube shape. So in fact, this is what a capillary looks like. The smallest blood vessels are just simple squamous epithelium. And we also find this tissue forming membranes. The parietal peritoneal membrane that lines the inside wall of your intestine is just a single layer of simple squamous epithelium. Then on page D4, we talked about simple cuboidal epithelium. Again, the ones with stars or asterisks, we want you to both know about them and be able to recognize them under a microscope. I had them out last time. I've got them out again. We'll be happy to put them out again on Wednesday. Remember, all the microscopes that I ever put out, there are always videos linked on my website, so you can watch them anytime you want. The simple cuboidal epithelium either looks like this or it's rolled into a tube shape. Where do we find it? All the glands of the body, salivary glands, thyroid gland, liver, pancreas, which are glands, are cuboidal epithelium. Where do we find cuboidal, simple cuboidal epithelium rolled into a tube shape? We find them in the urinary tubules, the renal tubules of the kidney. And if you looked at this last time under the microscope, and it's there again today, it looks really absolutely identical to this picture here. When you're looking at the microscopes, take your notes with you. And as you're looking through the microscope, read about it. I see so often students just looking in there saying, I don't see it. Do you know what it is you're supposed to see? All right? In other words, you need to read about it and understand what it is you're looking for. Furthermore, students will say, it's pink. Okay, I'm going to remember that it's pink. Actually, almost all of these look pink or purple. If all that you know is the color, it means you really don't know anything about it. The color is totally artificial. All of these have been dyed. All right, in real life, most cells of our body are transparent. They have no color. Now, I'm not going to trick you. I'm not going to put a slide out on a test that's a different color than the ones we're showing to you. I'm going to use the exact same slides. But if all you say is it's purple, it's pink, it means whatever you know isn't even real. You can use the color to help you remember, but really the important thing is the shape. The shape is real. So focus on looking at the shape of the cells. That's the part that's real. The pattern, the way they're organized, that's real. The color is totally artificial. All right, so try to notice the shape. Another tissue we said you didn't have to see under a microscope, but just know about it, is simple columnar ciliated epithelium. And that forms the inner lining of the fallopian tubes, or oviducts, and the cilia push the egg or embryo. On page D5, another tissue, all you have to do is just know about it, but you don't have to recognize it. The purpose of the mucus is to coat the inside lining of the stomach to shield it against the hydrochloric acid and enzymes. All right? Without that protective mucus covering, the hydrochloric acid and enzymes would just break down and kill all the cells. Another tissue, tissue number 5, which we covered last time as well, you do need to recognize that under the microscope. We had it under the microscope last time. It's on the microscope again today. Simple columnar epithelium with microvilli and goblet cells. Incidentally, I'm not going to take off if the way you write it isn't in the next. It doesn't have to be that exact order. In fact, I would tend to say actually simple columnar epithelium with microvilli and goblet cells. So as long as you're writing down the characteristics of that tissue.
Also, we have to
Simple.
write it after goblet cell.
Huh?
The whole sentence.
You've got to have all these words. You can't leave out any of the words.
It's not like a simple columnar epithelium. It's not.
Well, if you just write simple columnar epithelium, that you haven't adequately described this, because it has microvilli and goblet cells. So I just said it doesn't have to be in any particular order. So you could, as long as you could write simple columnar microvilli epithelium with goblet cells, you could write simple columnar epithelium with goblet cells and microvilli, but you've got to list all those characteristics to make it clear that you know that it's different from this tissue right above it, which doesn't have microvilli. This is the tissue we learned that forms the inside lining of the small intestine. And we looked on page D6 last time. We had microscope setup that looked just like these 3 pictures. This is really the macroscopic level of a cross-section through the entire intestine. These are called villi. Not microvilli, but villi. So this is made up of thousands of cells that form these macroscopic villi that you can see with your own eyes. If we enlarge this area, all right, and we had a microscope like this, now you can see these villi a little bit better, and you can even start to make out some of these cells. You can see there's a single layer of tall columnar-shaped epithelial cells, and when we look at it at higher magnification, we can see both mucus-secreting goblet cells and microvilli. If we enlarge this area right here, notice we see, just looking before we magnify it, we can see cells here and cells here. So if we enlarge it, here they are. So here's the cells and here's the cells, and you will see mucus-secreting goblet cells. The mucus has been artificially dyed blue. It's not really blue. I'm certainly not going to trick you and make it to give you a slide where it's green. I'm going to use the exact same slides. But again, try to look at the tissue, the shape of the cells, the vacuole that secretes the mucus. That's real, not the color. And we kind of have a fuzzy border right here. It looks almost out of focus. Those are the microvilli. And that's how it got the nickname brush border, because it's just, you can't make out, like if I was holding a hairbrush, it would be very difficult to make out the individual bristles. You just kind of see this fuzzy, looks like a brush. So it's called a brush border. You can't really make out the individual microvilli when you see it. All right, so that's, we had that out last time. It's out again today. And so let's learn another one. Number 6 on page D5. Page D5, pseudo-stratified columnar ciliated epithelium with goblet cells. Now, what's pseudo mean? Fake. False. So why is it said to be pseudo-stratified? And the answer is shown right here. When we look at the cells, and it is, when you look under the microscope, it's a little bit difficult to make this out. But when you look at the cells, they're not all the same height. Some of the cells are taller, some of the cells are shorter, right? Like this one. So because the cells are not all equally tall, it gives the false impression that it's more than one layer of cells. So it's called pseudo-stratified. It looks like, a fast look, you'd say, oh, it's multiple layers. But it's really not. It's one layer. It's just that the cells are not all the same height. So they chose to call it pseudo-stratified. It does have cilia. You will see the cilia. And incidentally, this is the only tissue that I've asked you to see under the microscope that has cilia. It's not the only tissue we're learning that has cilia, but it's the only one that we're looking at under a microscope. So if you can see cilia, you know it's this tissue, because it's the only ciliated tissue I've given you to see under a microscope. And you'll see also in blue the mucus of the mucus-secreting goblet cells. You will see that. So this is a very important tissue. You'd say why? Because it forms the inside lining of our entire respiratory tract. All the airways, the inside of the inner passages of our nose, the inside lining of our airways, our trachea, our bronchi, our bronchioles, is all this tissue. The purpose of the cilia, as we've talked about back in section B, is to push dust and foreign matter that we inhale, to push it out of our airways and keep our airways clean. And really, the mucus acts to warm and humidify the air. We also talked about that before. You'd say when? When we were learning about paranasal sinuses, back on page E3. If you look at what we wrote on the bottom of E3, we said that our nasal sinuses are lined by a mucus membrane to warm and humidify the air that we inhale. So a nickname for pseudostratified columnar ciliated epithelium with goblet cells is a ciliated mucus membrane. Now you have to write the correct full name, but if they ever, and we will see in the future, when we get to the respiratory system, they commonly, for short, will call it a ciliated mucus membrane. That's a shorthand way of saying pseudostratified ciliated columnar epithelium with goblet cells. All right, number 7. This is a funny name, transitional epithelium. So this doesn't have the typical names of simple or cuboidal or columnar. It's called transitional. Notice I didn't put a star. You don't have to see it under a microscope. Just know about it. This tissue uniquely forms the inner lining of the urinary bladder, the inside lining of your urinary bladder. And the reason why they called it transitional epithelium is because the shape of the cells changes. Think about this. If you had a balloon and you started filling it with water, as you fill the balloon with more and more water, what's happening to the wall of the balloon? You're stretching it. It's being stretched as you fill it with more and more water. As the urinary bladder, this sac that holds urine, as it fills up with more and more urine, it stretches the wall of the bladder. And on the inner lining, when the bladder is empty, it looks like this. And when it's full, it looks like this. Notice the cells get stretched. They get stretched. If I had to give a name, you don't have to know this. If I had to call this a name, when the bladder is empty, it looks like a stratified cuboidal. You don't have to know that. But as the bladder gets full and the wall gets stretched and the cells get stretched, they start to look like stratified squamous. So what did they decide to call it? Transitional. It transitions. It changes. That's what transitional means. All right, so that's uniquely found in the inner lining of the urinary bladder. Okay, just two last epithelial tissues on page D7. Two last ones on D7. Important ones, too. Stratified squamous epithelium. All right, so again, the naming is pretty straightforward. Stratified means multiple layers. And now, see, this is it. Now, when we look at these cells, the bottom ones kind of look more cuboidal. The top ones look squamous. Well, they had to name it something. And they chose to name it based on the appearance of the top cells. So since the top layers of cells are squamous in shape, they called it stratified squamous. You have to call it something. So that's what we wrote here. It's named according to the appearance of the surface layer of cells. It's located in areas of the body subject to abrasion because this is a pretty tough tissue, multiple layers of cells, so it's pretty tough. And there are actually two subtypes of stratified squamous epithelium, and you have to not only know about both, but you have to recognize both types under the microscope. So there is what's called non-keratinizing stratified squamous epithelium and keratinizing stratified squamous epithelium. Let's start with the non-keratinizing. The non-keratinizing is called that because there are no dead cells. They're all alive. And this is the type of tissue that makes up the inner lining of your oral cavity. You'd say, what's that? The inside of your mouth. Also the inside of the anal canal and the vaginal canal. So in other words, you'd say, well, like, I don't know what you're talking about. So I'm touching the inside of my cheek. The inside of my cheek is made up of this tissue. It is made up of non-keratinizing stratified squamous epithelium. All the cells are alive because they're all surrounded by, inside our mouth, we have saliva. We got fluid. So they're all living cells. And so this is the skin. They're on the inside of your cheek. Inside of your cheek. Inside of your mouth. The oral cavity. Also the inside around the anus and the vaginal canal. All right, it's pretty tough. Now, a second type is called keratinizing stratified squamous epithelium. Keratinizing, in contrast to non-keratinizing, means there are some dead layers on the top surface. Dead layers on the top surface. This is the tissue that makes up the outer epidermis of our skin. So this skin right here, a skin all over your arms, all over your face, outside of your face, all over your body, all right, that's keratinizing stratified squamous epithelium. The top layers of cells are dead and sloughing off. Why are the cells on the surface of our skin, on the surface of our body, why are they dead? Because they're in contact with air. We've talked about that before. Cells can only be alive if they're surrounded by fluid. So these are dead cells. Another difference you're going to notice between these. So if this is the non-keratinizing stratified squamous epithelium, like on the inside of your cheek, and this is the keratinizing stratified squamous epithelium, it's right here, but these top layers are sloughing off. They're kind of coming off. Another difference you'll see is that in the keratinizing stratified squamous epithelium, you're going to see a brown color. That brown color is real. That is the brown-colored protein melanin. We have melanin in the skin on the surface of our body. The purpose of the melanin is to shield us, to protect us from harmful ultraviolet rays. That's the purpose of it. Some of us have more melanin, some of us have less melanin. That's genetically determined, right? But no matter how much melanin you have, if you repeatedly go out into the sun, day after day, what's going to happen to the amount of melanin your skin cells have in them? It's going to increase. That's called tanning. right? They think that tanning is a darkening of the skin because it's our body will produce more melanin to shield ourselves from that harmful ultraviolet rays. So you will see that brown color in this type of skin. You will not see it. You will not see it in the tissue that's found on the inside of your cheek, because on the inside of our cheeks, it's pink. There is no color. That's just the blood. You should see both these tissues under the microscope and be able to recognize them. So we have the skin on the outside of our body and with the skin on the inside, like of our mouth.